Today, I'm going to walk you through the Yield by Design Investigating Hybrid Seeds lab from Kansas Corn STEM. Hi, my name is Lacey and I'm a lead teacher for Kansas Corn STEM. I've been teaching science and biology for the last 11 years, and this year we're also throwing in a little agri-science. So today we're going to go through the hybrids lab that Kansas Corn STEM offers for us, and it comes with a lot of amazing things. But before we get started there, I want to walk you through a little activity you can do with your students to activate their prior knowledge of genetics and appearances. Here we go. Who is this man? It can be really any picture of a person that you would like to do. This is one of our staffers. So take a good long look at this guy. What would you need to identify this person again? What features would we look at? And you can encourage your students to write down what these features are. Give them a good 2-3 minutes to do it. Then you could have them do something else for 2-3 minutes. Get up, stand up, walk around, go get a drink, and then have them come back. Now, which one is he? Have your students walk through and think about what identifying features they saw before and be able to identify the man. Hopefully they can. If you can't, here's a hint, he's in the back of the truck. Now we're going to take this same approach, but with something a little different. Check out this plant. Don't forget to write down all those major features. What are some of the features that you can see of this plant? Now here your kids are going to start complaining, but have them go through and talk about what are some features that we know that we could write down. Think about the number of leaves, the fact that it's tasseling, does it have any ears, and go from there. Now which plant is it? Hard to tell, right? As we look through this field of corn, all of those plants look so similar. Their traits are so similar. How can we tell which one is the individual that we looked at before? Well, that really brings us down to understanding the differences between organisms. There are micro differences and macro differences. The first picture we looked at, we really focused on macro differences, those large features that we can easily see to distinguish between individuals or items. When you're doing your DNA unit, a lot of times we talk about genotype and phenotype. This would be the phenotype section. But when we look at other organisms besides humans and animals, sometimes it's harder to distinguish between macro and micro differences. So we have to look at those macros. What could be those examples? If you've already pointed out the phenotype, hopefully now they'll look at the genotype. So what are we going to do? How do we figure out how that plant is different than the rest? Well, we got to look at those micro differences. And the best way to do that, hopefully your kids can figure it out as well, is to look at the DNA. Comparing DNA can be a hard process. And because it is so microscopic and kids can't really see the molecule, it's hard for them to understand. So this lab is going to walk you through 2 ways to do that. One in a lab setting, and one is an activity that you can do during the lab. Whoops. So comparing DNA is pretty simple. We've got some things to help you out. If you go online to Kansas Corn STEM, you can find that we have a lab that goes along with this. And in our lab, we have some DNA sequences. I know they're very tiny and you can't see the letters, but each one of these has a DNA sequence of different A's, T's, C's, and G's. They're numbered at the front, 1, 2, 3, 4, so that you can disaggregate amongst the groups of students, or you can give one individual an entire pack of all 4. What we're going to do is give them some options. They have 2 choices. They can either compare letter by letter by letter. Here's where the groans come in. This is a lot of letters. So what's the shortcut? That's going to be what the kids always want to know. What is the shortcut? And that's where our next step comes in. There is another way. So as we go through and look at this, we can use something called a restriction enzyme. A restriction enzyme is a molecular enzyme that goes in and cuts DNA after a specific DNA sequence. Here's some examples. So we're going to use 4 make-believe restriction enzymes. Students are going to go through their sequence and look for these 5-letter combinations. Anywhere where they find these 5-letter combinations, they're going to go ahead and mark on their piece of DNA sequence. I use 4 different colors, red, blue, green, and yellow, to help my students separate these out. To do this, really all you need is the sequences and a couple of markers. Have your students literally go in with each marker and identify where you see that sequence. So here, because I'm using my blue marker, I'm going to look for the sequence of TAGCG. And I have found that sequence on number 4 right here. So I underlined it in blue. Hard to see, but it's there. Okay. Another alternative you can do is if you have colored highlighters, you can do the same thing with colored highlighters. Here you see the highlight, might be easier for your students to see. As you go through, you'll repeat that process for all 4 colors. The next thing you're going to do is have your students actually cut their DNA right where we have that mark. So in the end, instead of having one piece, you're going to have multiple. Simple way to do that is to have them pinch it off right there or use a pair of precision scissors, either way. So now what I have is multiple fragments of DNA that have been cut by my restriction enzymes. This is the key to make this magic work. Those restriction enzymes do all of the heavy lifting for us. They're programmed to cut at those specific sequences. So now, anytime they see them, they're going to go in like that precision pair of scissors and cut that DNA into fragments. So depending on where and how many times that restriction enzyme might or might not be seen in the sequence, now we're going to have different sizes of DNA fragments. We can then compare those fragment sizes to figure out if 2 samples of DNA came from the same individual. How many fragments do your students have? Do all of their fragments match up? Are some larger and some smaller? How would we figure this out in a lab setting? That's our next part. Today, we're going to be using the MiniOne system that is available to you through checkout through Kansas Corn STEM. Okay? If you are interested in checking these systems out, please make sure that you go onto our website and go to the Request Materials tab. This system is very easy to use. If you've done electrophoresis before, which is the system that we're using today, this will look like a very simple version of all of the things that we have done in the past. But instead of having to go through all of the prep, it's very, very simple. The items that you will need for this come in the kit that you can check out from Kansas Corn. We have the MiniOne carriage, which is our power source. We have our tank for our electrophoresis gel. We have a viewer that allows us to illuminate the gel. You will get access to some of these wonderful microcentrifuge tube racks. You'll have an appropriate gauged micropipetter and a gel form. All of this works together to make a pretty slick lab. So here we go. We're going to go ahead and get started. The first process to any electrophoresis lab is to cast the gel. When you get the kit, there will also be a wonderful picture guide that helps walk you through and your students how to cast these gels. In the past, sometimes this would be something that teachers would do independently because it takes so much time. But now, within 20 to 30 seconds, your students can make their own gels. Here's how. The kit comes with a tray that looks a little like this. This tray allows us to be able to change the number of wells that we want in our gel. You can see on one side we have a smaller well that allows for more samples. The other side we have a larger well that allows for fewer. Today we only have 5 samples, so we're going to go ahead and use the larger wells down here. The other great thing about this is the tray itself. It has electrophoresis trays that look like this. In the past, we would always have to tape or add rubber bumpers to the edges of this. But now, because it has a molding tray, it slides right in. We pop in the comb, and we're ready to go. In your consumables kit, you will also see that you get these TBE gel cups, if you want to call them that. They will already have the agarose with the TBE and with the dye already in here. So students don't even have to touch any of those chemicals. You peel back the top, microwave it for about 10 to 15 seconds until it's completely melted, and you're ready to go. Peel off the top. Make sure your comb is in the right position with the larger well down and pour it straight in. In about 10 minutes, this will be completely set up and allow you to be able to put your gel in place. Here's one that I've completed for you. So now that it's set up, I can actually tap on it and it feels like that very firm Jell-O like we've talked about before. So to release it from the mold, all I do is pull straight up on the comb and pull up on the edges of the tray. Now there may be a little of the gel that's seeped underneath, but all it takes is one quick finger swipe on either side, and you're ready to go. One thing I always point out to my students is there is no plastic on the top and bottom. So it's very important that as they hold these, they're holding them with a little brace below, just like this. So now what we're going to do, we can set this aside. And by the way, did you notice that each one of these trays allows you to cast 2 gels at once? So you could share a tray between 2 groups of students. You'll need 2 gel cups, but only 1 tray. So now that we have our gel cast, we're going to go ahead and load it into our system. Again, I'm just cleaning off the edges to make sure it's good and clean. And I'm going to add it into the tank. It slides right in. In case you wanted a little bit more detail about what that gel looks like up close, there we go. Once the gel is loaded in, everything is working on the tank and everything is in there correctly. You will see that in the tank, I have 2 electrodes that move all the way through. This is what's going to be supplying our current for the electrophoresis process. Right now, all I have in here is my gel. I don't have any buffer in here. So that's going to be our next step. Here it shows us turning the light on before we put the buffer in. However, I'm going to suggest we do this maybe a little bit of a different way. With my students, I find the risk of having them pour the buffer in the carriage to be too much. So what I try to do is have them take the tank out and put it in front of them. The TBE buffer comes in a concentrated bottles with very specific instructions on how to dilute it for your students. Each group only needs about 135 to 150 milliliters of the buffer in the tank. I encourage the students to pour the buffer only on one side. That allows the buffer, as it washes over the gel, to force any air bubbles out from underneath the gel and make sure that we have a nice connective surface. This process is all about ease and slow patience. We want that buffer to move its way all the way across and completely cover the gel. Right now it's covered the top of the gel, but it's also percolating into the other side of the tank. And I want both sides of the tank full, as well as a very thin layer on the top of the gel. Now that I have that accomplished, I'm ready to go ahead and put this into the carriage. Remember these little electrodes that I showed you before? These electrodes here match up with some gold rivets that are actually in the carriage. That tank will only go in here one way. Making sure that I have my wells pointing towards the negative part of the carriage and not the positive. It is marked on the carriage right here, negative and positive. If I have my carriage turned on, Now the blue light is going to illuminate and we will be able to see the wells in the gel. If it does not turn on, just push the button. From here, we're going to go ahead and load the gels. So take a look at the image that you see on the screen here. Now that everything is in the gel, all we have to do is add our wells in. This process is going to involve your students micropipetting DNA samples into those teeny tiny wells that we just made. Sometimes this can be difficult for students. Make sure that if you haven't already done so, practice using our pipetting lessons before so. The micropipetter that you get in the kit is going to look a little bit like this. Okay. It has an adjustable dial that has 3 numbers. The first 2 are black, the last one is red. This micropipetter goes from 2 to 20 microliters. For our lab today, we need 10 microliters. So to adjust this, you can either twist the top knob or you can use the wheel here in the middle. We're looking for 10 microliters, so it should read 1, 0, and then another 0 in the red. One thing to remind your students is that when we use micropipettes, especially with DNA samples, we need to make sure we use a fresh micropipette tip every single time. Each group is going to need their own pipetter and their own set of tips. Another helpful tip is to give them a container to get rid of those tips in. It can be as simple as a solo cup, or if you don't have that, use a beaker and wash it later. To load your pipette tip, you simply hold your micropipetter with the little nubbin over your finger, and you're going to push straight down. Now we've got it on there. As a teacher, your DNA samples are going to come in your consumable kit and look a little something like this. Each one has a color-coded top, and I number the top so I know which one I need to make sure gets in each place. I go ahead and aliquot out all of the samples into a smaller container that also comes in the kits for my students. So my students will have a rack that looks something like this with numbers 1, 2, 3, and 4. Each one is in a different color of test tube, but most importantly are those numbers. This indicates the order at which they're going to get put into the wells from left to right. To do this, there are a couple of things to keep in mind. Steady hands are very important because this sharp tip will go all the way through that gel. If it does that, all of your sample gets released underneath the gel and it won't travel through the gel appropriately. Place the carriage closer to your workspace. Open the tip. Go ahead and depress the plunger. Put it down in and withdraw the sample. Now that the sample is completely used up in this, I could throw it away. However, I like to have my students cap it and put it back so I can make sure they actually drew up the sample they were supposed to. From here, we're going to put it in the very first well. This is the tricky part. There are 2 tips I have here. Number 1, use your elbows, and number 2, steady and slow. If you put your elbows down on the table, and be able to take this, you can go straight up and down, all the way into the well, depress my plunger all the way down, and holding it down, pulling my hand straight up. If I release the plunger while it's still underneath the level of the liquid, it's going to suck all of that liquid back up into the micropipette tip. That's a no-no. So make sure when your students are done loading the sample, there should be nothing left in this tip. If there is, encourage them to do it again and make sure they hold that down in the same well. From here, I can go ahead and release this and load the rest of the 5 samples. Always encourage your students to take turns. Lab work isn't just for the few, it's for everyone. This is especially the fun part, so make sure that each student gets a chance to pipette. After you have all 5 samples loaded, you will be able to see the dark color in all of those wells. For today, we're just going to load the 1. Now, we need to go ahead and talk about running this race. We're going to take our view hood. It has some magnets that are right here that engage with the carriage. From here, you will see the green light turn on. Now you can make sure that green light is on and everything is running. Students are encouraged to peek through this hood about every 5 minutes to make sure and see that the progress is working its way through. Now that we need to wait about 20 minutes for this DNA to do its work, we need to talk about what's actually happening here. Well, really we have a DNA race. Remember, we cut up all of that DNA into tiny pieces. Some are bigger, some are smaller. I like to talk about this as it's a race. When I was in school, we had what they called the heavyweight 4x1, which means you had your throwers and your biggest guys running that 4x1 right next to your sprinters. Which one do you think is going to end up as the winning team? The slower guys, the heavier guys, or the faster guys? Who's going to end that race quicker? In this example, what you can do is have your students pair off or get together in larger groups. Have some small groups, 1 or 2 students, and some larger groups, 5 or 6 students. They must link arms and basically run through an obstacle course. In a classroom, this can be created by running through rows of chairs, or if you have access to your gym, go ahead and put some obstacles out there for them to run through too, because that's really what makes it fun. Generally, at this point, as students are running through, they'll find the ones that have 1 or 2 people can go much faster. And so by the end of the time, only give them about 30 seconds, you'll see that some of them have made it further across the obstacle course than others. And that's exactly what's happening here with our DNA. Okay? So DNA has a negative charge, and they are just like the runners in this race. They are going to run to the positive side or the finish line of our racetrack. That electrical charge that is moving through the phoresis gel right now is going to be like a starting position for our DNA. And once we hit that green button to electrify the gel, we are making those DNA samples run their race. And as we give them the exact same amount of current, the shorter ones are going to move faster down the lane or down their part of the gel. The longer ones are going to move slower. So as you can see here, I have a couple of examples of other pictures of electrophoresis. The top being where the wells are, the bottom being the finish line. So the ones that are closest to the top of those pictures are going to be your biggest fragments of DNA. The ones that are farther away will be your shortest. And that's how we can compare our DNA, by looking at the bands. If the restriction enzymes have cut the DNA in the exact same sequence of bands, we know it has to come from the exact same organism. If the restriction enzyme cut them in different bands, then we know it came from different organisms. The more restriction enzymes you use, the more bands you get, and the more variety that you can see in those individual DNA sequences. You can have your students finish up their modeling activity by using a graph. This graph really helps to simulate exactly what's happening here with my larger numbers of my larger base pairs, which are just the letters on here at the top, and my shorter ones at the bottom. Make sure that you reference this online as well. Every 5 minutes, have your students view through here and make sure that things are moving. If things aren't moving, they should immediately raise their hand and have you come check. Some of the most common things that can happen is they can punch a hole through the gel and the DNA sample could be running out the bottom. It could be that the gel has accidentally slid and is touching an electrode, or it could be simple like it's not turned in or it's not plugged in. Once you've got those 20 minutes of data done, your students can actually take their cell phone, put it right up next to this viewfinder, and snap a quick pic. That is the beauty of this. Also in here, you will see, as you look down, that there is a wonderful little ruler in there. So if you want to use Logger Pro, you can also use their electrophoresis analysis tools to help you with this as well. Now that we have results, have your students go through the final questions of the lab. So which plants have the same traits? Which bands are the same? Fill in the tables that you see there and have them come up with a concluding question. And by the way, just in case you need it, here are the results. Thanks so much.